Hello and welcome to another interview. Today we'll be talking with Dr. Rachel Whaley. For this episode, we'll be talking about wheat breeding. Could you introduce yourself?
Hi, everyone. My name is Rachel Whaley. I am currently a PhD student at the University of Guelph. My research project is focused on improving winter hardiness and winter survival in winter wheat varieties grown in Eastern Canada, mostly in Ontario because the production range is currently restricted because of winter survival issues. I was originally in the lab of Dr. Ali Navabi before he passed away in 2019, and I transitioned over into Dr. Eric Lyon's turf lab, and I'm also co-advised by Dr. Joe Colasanti in the molecular biology department. So we started out by trying to figure out why certain varieties in Ontario survive the winter better than others, and the goal for increasing our understanding of this trait is we're looking for traits that can be incorporated into the plant breeding program and actively selected for in order to improve winter survival and winter hardiness. The problem is that most of the literature in terms of winter survival is based in Western Canada, where they have a completely different growing environment. So in Western Canada, the main limiting factor for winter survival is lethal temperature. It's not uncommon for the air temperature to reach negative 40 degrees over the winter in Alberta. Now, that's an environmental condition that is not or very rarely encountered in Ontario. Our winters are more characteristic of having a moderate cold temperature. We can get down to negative 10, negative 15. But we have more freeze thaw cycles. So it's not uncommon for certain time periods of our winter to be above freezing. We can have quite a bit of winter precipitation, which means that we have water laying in the fields, which is a completely different abiotic stress than cold temperature. And there was a graduate student who just graduated from the turf lab, Jabir Husseini, who was focused on low temperature flooding and ice encasement. So back to what we're doing in my research, we're looking for these traits of interest that can be actively selected for in the plant breeding program. The problem is that we don't understand winter survival in Ontario winter wheat at all because our understanding is based on Western Canadian wheat. To start out with, we continued on the work of Andy Chen, who was a master's student in the wheat lab. And he had found that to survive in Ontario, winter wheat varieties needed to be strong winter types, which means that they have 3 copies of the vernalization A1 gene. And that's the gene that really controls the difference between vernalization requirement and having no vernalization requirement, or winter and spring type. There's also another cold tolerance locus that's very closely related to vernalization A1 called frost tolerance A2. And if it had a tolerant haplotype at that particular gene, it was considered extremely winter hardy and it should survive well in Ontario. The problem is that when you looked at this group, most Canadian winter wheat already was fixed for this genetic combination, and almost all of the Ontario varieties fell in this particular group. So the assumption was that there was something else missing that could explain the significant significant variation in winter survival, even in this elite population. Unfortunately, when we redid the GWAS, we didn't find anything conclusive. We did find a few loci that were associated with different types of abiotic stress, but our general result from that particular, our general conclusion from that particular result was that looking at winter survival as an umbrella term for all of the different environmental conditions experienced in Ontario was probably not a good idea. And we should be looking at all of the various components that contribute to winter survival individually. Next, we were trying to find differences between the non-hardy variety Gaines, which is a 3T variety, but it's native to Washington State and Oregon, where it's a very mild climate during the winter. and Branson, which is a representative Ontario variety that was widely grown across Ontario for 10 or 15 years. It's starting to taper off and be replaced by more modern varieties now. And we're still interpreting the results of the RNA-seq experiment, but what we found was the longer Branson and Gaines were exposed to cold acclimating temperatures, so this is 4 degrees Celsius, so not freezing, was Gaines seemed to be out of control. It had increase of reactive oxygen species, things that were markers of extreme stress that could lead to cell death and even plant death. Whereas Branson seemed to be in a form of stasis. It downregulated cell cycle and genes involved in duplication of the genome and cell division. So we're still exploring that, but it seems like Branson does have a very distinct way of dealing with extended periods of cold temperature compared to a non-hardy variety. We also looked at physiological traits associated with increased winter survival, and these were all things that are very common in the Western.
Interrupt quickly?
Yeah, of course.
So for context sake, there is a sort of problem of acclimatization, and Branson has the traits that allow it to acclimatize to longer periods of cold for much quicker and much longer.
So I wouldn't say that it can adapt to periods of cold for much quicker, but it can withstand cold temperatures for longer without losing its hardiness. The most generic way of describing this would be that Branson has some sort of local adaptation to Ontario, which makes sense because it was developed for this particular environment, whereas Gaines, which is a variety from the West Coast of the United States, is not regionally adapted. So we're trying to figure out what the regional adaptation is that Branson has.
Thank you.
So when we look at our physiological data, there's really not a lot of differences in Branson and Gaines. They seem to sense temperature around, or they seem to sense cold stress around the same temperature. They seem to have the same level of hardiness, so they can each withstand about negative 13 degrees Celsius once they're fully acclimated. The key difference is that Gaines seems to lose its acclimation sooner, so it starts to have a reduction in its cold hardiness sooner than Branson does. And we're still trying to figure out why exactly that is, and if that's the key difference between surviving in Ontario and not surviving the Ontario winters. And I should go back and say that my project specifically is focused on cold acclimation. We're not looking at the various other factors that contribute to winter survival in Ontario. Winter kill in Ontario can be caused by a whole host of things. So if you're in Southern Ontario near Essex County, where you don't have snow cover, you can have cold-induced desiccation from north winds blowing across your wheat fields. Areas that have clay soil can experience frost heaving, which physically damages the root system and can push the crown above the soil surface where it's exposed to the air temperature rather than the soil temperature. If you're in an area that has prolonged snow cover, you can have apoxia. So there's not enough airflow getting to the plant underneath the snowpack. Other areas, you can have snow mold infestations if you have snow cover for a long period of time. In Eastern Canada, ice encasement and low temperature flooding is prevalent. Although when you look at the turf and alfalfa literature, ice encasement and low temperature flooding is a much more widespread Ontario-wide issue. And lethal temperature really isn't an issue until you get into Northern Ontario, where we really don't have a large acreage of winter wheat production anyway. And then the biggest one that really hasn't been studied sufficiently is we get these thaw refreeze cycles. So we can have periods of time where even in the winter, we can be above 10 degrees Celsius. And it's well known in the literature, at least in the Western varieties, that if you have an air temperature higher than 10 degrees Celsius, you will rapidly lose your cold hardiness.
So if I'm getting this correct, then please correct me if I'm wrong. There's a sort of limitation of cold hardness where that cold hardness only lasts about a certain period of time before it starts tapering off.
Yes. Yep.
And you're studying how to lengthen that cold hardness period. Is that correct?
We are trying to figure out if a longer cold hardiness period will allow the Ontario varieties to survive better. The whole goal of the grant that was funding my project was to increase winter survival in Ontario winter wheat. The problem is that there were a lot of assumptions based on a completely different genetic population, a completely different growing region. And no one's really looked at what makes Ontario winter wheat survive the environment that it's currently growing in. So my project is much more just trying to understand what our wheat encounters and how it manages to survive what it encounters in Ontario. And because there are so many factors, we were limited to looking at just cold acclimation and cold hardiness specifically. And there's other projects being spun off of this idea that are looking at all of the other environmental factors that can influence winter survival in Ontario.
So with this sourcing in play, if acclimation is permanently controlled, if the survivability of the wheat is permanently controlled by acclimation, theoretically, if you increase that amount, that would increase the survivability, correct?
That was the working theory. So Branson is the representative Ontario variety that we're using, but it's really not been evaluated in the literature before. If you look in the literature, most of our knowledge of winter survival, cold hardiness in wheat is based off of a variety named North Star, and it's a Western Canadian variety. It is extremely cold hardy. It can withstand temperatures of negative 25 degrees. But when you grow North Star in Ontario, its survival is significantly lower than the varieties that are commercially grown in Ontario. So in the case of North Star, having superior winter hardiness did not equate to superior winter survival in Ontario.
All right, I'm going to go through a quick series of questions. Well, maybe not quick, but first question is, could you theoretically study the physiology and the genetics of Bentley, I believe is what it's called, correct?
Branson.
Branson's, thank you. If you could study the Branson cultivar, you could conceivably get a full genome analysis and figure out how each one works and study it biochemically, and you can maybe create a full list of all things that make this plant suited for Ontario.
That
Could
is
that
the
be a
goal.
spin-off project?
That is the goal. We're trying to create a profile of Branson from the various experiments that we've conducted for my thesis. So with the RNA-seq experiment, we're trying to identify different genetic pathways that are at play during a 98-day cold acclimation period at 4 degrees. So what we've done is we're using time point 0 as our control group, and then we're looking at the differential expression at day 35, day 63, and day 98 under periods of cold acclimation. And we were also doing that with Gaines, which is a well-known non-hardy variety in the literature that does quite poorly when grown in Ontario. So we're trying to identify what makes Branson unique, what particular strategies it uses to survive this particular cold acclimation period. And when it comes to physiology, it's quite common practice in the literature to characterize the threshold induction temperature, its hardiness at 28 days of acclimation, the time it takes to become vernalized, because a plant that is reproductive has a harder time reacclimating after periods of warm temperature over the winter. So the thought is that a variety that takes longer to vernalize may have a better survivability because it can reacclimate after periods of thaw and refreeze cycles. We're also looking at how the varieties accumulate carbohydrates like fructans, which are known antifreeze proteins. We were also planning on looking at abscisic acid, which is the plant stress hormone that regulates all the different plant responses to abiotic stressors. Unfortunately, due to COVID, we were not able to go forward with that particular experiment, though.
I have a question. Is there any chemicals that inhibit the phasing out of cold hardness? Like we have occasional periods where it's 10 degrees Celsius, but still winter. And so there has to be a way of inhibiting growth. Is is there any studies regarding that going on?
That's not something that I've looked at specifically. It is pretty well known that plant hormones have quite a bit of control over the cold acclimation or just cold responsive pathways in general. Like I said, abscisic acid. I'm not sure if there's any commercially available plant growth regulators that could be applied to the field. The biggest problem with trying to recommend something like a plant growth regulator to a crop like wheat. So in Ontario, wheat's not a very profitable crop for farmers to grow. Most Ontario farmers prefer to grow corn and beans in rotation, and it required quite a bit of research to convince farmers that they should incorporate wheat into their rotation just to have a boost in yield for their other crops. Is not very profitable, so trying to convince farmers to spend more money on a wheat crop is challenging, especially when you have crop insurance. So if you have a bad year and your wheat crop is lost, it's not that big of a hit to just plow it under and plant soybeans.
Makes sense. So, do you know of any other means of improving cold hardiness? Any genetics around freeze thaw reactions, like stronger root systems, like maybe that may play a role in cold hardiness?
That's definitely a theory. My advisor, Dr. Lyons, he's a turf physiologist, so he's constantly looking for physiological traits, physiological differences that could be at play that could give certain varieties an advantage for winter survival. That's definitely something that still needs to be explored. So the last time that Eastern Canadian winter wheat was really evaluated in the literature was in the 90s. So there's quite a few opportunities for further research in this particular area.
So, how often does the freeze thaw damage happen within wheat, and specifically, in what conditions does death by absolute loss of cold hardness happen? What, on average?
Also, that's also pretty hard to quantify as well. So, really the only metric that we have for when winter survival has been an issue is when farmers claim crop insurance on their wheat fields. The last significant year that we had was 2018 and 2019. And unfortunately, we don't have the data to make any concrete claims as to what environmental condition actually caused such. I think it was a 29% reduction in harvested acres of wheat and a 8 or 9% reduction in overall harvested yield. Those are really the only metrics that we have from year to year is how many acres were harvested and what the average yield was. It's thought that that particular year, it was very cold and very damp well into the spring. So something like ice encasement and low temperature flooding may have been responsible for that particular loss. As far as I know, LT50, so LT50 is the metric we use to quantify hardiness at a particular time point, is not regularly observed in the field in Ontario. They do have a monitoring system in Western Canada, and they quantify it in winter kill days. So the number of days that the air temperature or soil temperature drops below the predicted hardiness for a particular variety. And The last time I looked at that particular website, whenever they had a period of warm temperatures followed by a reduction of air temperature, like back into the negatives, the number of winter kill days did increase.
Thank you. Now I have two more questions. The first one would be, let me think how to ask this question properly. Have you noticed any patterns on how the research is going and what direction all the answers seem to be funneled into? Like, does it seem to be like they're all the data is pointing to one specific direction? And if so, what is that direction and what do you expect will happen after your thesis is done and research has been analyzed to the fullest?
Yeah, so like I said, we're still analyzing the RNA-seq data, but the interesting thing that we're seeing is that Branson seems to be in a holding pattern, especially after 98 days of cold exposure. So there, if you look at the non-hardy variety gains, that's not performing very well in Ontario. If you hold gains at 4 degrees Celsius for 14 weeks, it starts to upregulate things that are associated with reactive oxygen species. So that's an indicator of cellular stress. We're not seeing that in Branson. So Branson seems to have a way to stay on top of the accumulation of reactive oxygen species. And it has a significant downregulation of genes that are involved in cell cycle progression. So if you look at other species like corn, they have also observed this sort of holding pattern when varieties are exposed to periods of prolonged cold stress, where they're just trying really hard not to have any sort of cell division to reduce the amount of energy that the plant requires. And that seems to correlate. We still need to do a little bit more analysis before we can make any concrete claims here, but that holding pattern seems to correlate with prolonged cold hardiness when we observe the LT50 of the same plants treated in the exact same way. So it'll be really interesting to see if that is what allows Branson to survive a little bit better than Gaines does. As I said, there are quite a few other environmental conditions that influence winter survival in Ontario, and it'll be really exciting to see further research that explores all of those different abiotic stresses. So instead of evaluating for winter survival in a plant breeding program, possibly looking at indirect selection methods. So looking at components of winter survival, components of like all of the different environmental factors that influence winter kill.
Thank you. So if Benson has a longer holding period where it can stay dormant longer without having any damage, and that's one of the main factors that contribute to cold hardiness, what would you try to come up with a screening method to determine longer varying degrees of holding periods and use that as a means to screen for that trait? And if so, what type of screening method would you use to make that happen?
So the answer to that question will vary depending on the specialty of the person that you're asking. So because I'm in a physiology lab, looking for particular physiological markers like is the acquisition of fructans or the amount of fructans that they are holding on to at 98 days, or if we can quantify the number of reactive oxygen species, if we can accurately perform LT50 data so we can have a metric of hardiness to compare varieties against. All of those things could be used as adequate screening markers. But when you think of it in the context of a plant breeding program where you're screening tens of thousands of lines across numerous multi-environment trials, the amount of labor required to accurately quantify all these different markers that you could use um, that are associated with cold hardiness or maintaining cold acclimation status, it becomes unrealistic to try and implement those in a plant breeding program. So you've already talked to Dr. Alexandra Fitch, and her project focused on genomic selection. So indirect selection, picking traits that are strongly correlated to winter survival, but are easier to quantify, and putting those into Alex's genomic selection model is probably the best way of implementing these screening methods in the context of a plant breeding program. We definitely need to spend more time trying to find easier ways to quantify all of these different traits. So the fructin analysis is quite a lot of man hours, LT50. You're limited by the amount of freezer space and acclimation space you have. So there's a lot of bottlenecks that definitely could be worked out to make it streamline a little bit more. But it definitely needs some sort of genomic selection or marker assisted selection model to streamline it into a plant breeding program.
Thank you. And I think that about covers everything. I'm just going to stop the recording. But first, I want to say thank you for your time and your participation within this podcast.
No problem at all. I always enjoy the opportunity to talk about my research.
Thank you for watching this video. If you enjoyed this video, please like and subscribe. If you really enjoyed it and think you can donate, you can do so at Buy Me a Coffee, link in the description below. Thank you for watching.